Okay, a couple of quick announcements. Don't forget that you can get some optional points today for the biostatistics and human disease talk today at 4:30 here. I will be in another meeting. Dr. Morton McCombs will be here with the sign-up sheet. So if you attend, please sign the sign-up sheet. And then some news you probably will appreciate. Dr. Morton and I have been talking, and the lab presentations were scheduled the week after you get back from spring break. That doesn't really leave much time for working together and getting things set up. So what we've decided to do is we're going to flip two labs. The lab that we scheduled for the week that we get back from spring break was the genetic transformation, the P-Glow lab. No, sorry, the enzyme, and then the P-Glow lab was the next week. We're going to flip those two. So when you come back, you're going to do the P-Glow lab, and that will give you another week after you get back to work on your presentation. So they will be 2 weeks after break. Okay? No objections, I'm assuming? Okay. Okay, let's do a little quick review before we continue on. So when we started introducing photosynthesis, we kind of broke it up into these two black boxes that we indicated were light-dependent reactions and light-dependent reactions. Where do the light-dependent reactions take place in the chloroplast? What are the two major functional parts of the chloroplast? What's that fluid-like, filled material? Okay, there's a stroma, and then embedded or floating around, if you like, in the stroma are what? There are membranes that we call thylakoid membranes, okay? So which of those two locations is the site of the light-dependent reactions? 50-50 shot. They're associated with the thylakoid membrane. And then by default, the light-independent reactions happened in the stroma. What's another name for the light-independent reactions? Well, okay, we used to call them the dark reactions, and that's not a great name, so we're going to just forget about dark reactions. It's a cyclical process, Sam. This is the Calvin cycle, okay? What are the inputs associated with the light-dependent reactions? Or if you like, what are the substrates of photosynthesis? Okay, the main substrates of photosynthesis are carbon dioxide and water. Only one of those is associated with the light-dependent reaction. Which of the two is it? Water. So during the light-dependent reactions, water molecules are being split. And what is a waste product from splitting that water molecule? Oxygen. So when two water molecules are split, they each release an oxygen atom, and then those two oxygen atoms are unstable, and they immediately combine to form an oxygen molecule. Okay? So that leaves us with a bunch of hydrogens left over. Okay? Where do those hydrogens go? They reduce something. What do they reduce? A coenzyme. The coenzyme is kind of the cousin of NAD, NADP. So we reduce NADP. There is a pigment or pigments that are involved in absorbing the light energy in the light-dependent reaction. Those are chlorophylls, and we'll talk about some other related pigments to chlorophylls as well. So when those chlorophyll molecules absorb photons of light energy, what happens? What happens when you flip open your solar calculator? We get a current, right? We get kind of an electrical current. And in this light-dependent reaction, those photons stimulate, if you like, or excite, we'll see in a minute, chlorophyll pigments. And those electrons start passing through an electron transport system. Okay? And as a result of electron transport, what happens? We form a hydrogen ion gradient. So we build up hydrogen ions on one side of the thallipoid membrane compared to the other. And then what can we use that hydrogen ion gradient to do? Make ATP, right? The process by which ATP is made is pretty much the same as it was in cellular respiration. We call that process of making ATP in cellular respiration oxidative phosphorylation. Because remember, the oxidation of things like NADH and FADH was what was driving the electron transport chain. That's what's driving this electron transport chain? Are we oxidizing NADH or FADH? No. What's driving that? Sunlight energy. Okay? So this particular process of making ATP is an oxidative phosphorylation. It is called photophosphorylation. So the sunlight energy is providing the impetus, if you like, to drive this electron transport chain. And as that electron transport chain is functioning, we generate the hydrogen ion gradient and we make ATP. Okay? So here are our three major products of the light-dependent reaction. Any CO2 involved yet? Nope. So, what happens in the Calvin cycle? Carbon dioxide is now involved in the Calvin cycle. And ultimately, as a result of this Calvin cycle, glucose is made. Since this cycle has to constantly regenerate itself, it would take feeding in 6 CO2s to make one glucose molecule. Okay, because remember that's C6H12O6. So all of the carbon that ends up being all the carbon in glucose comes from where? Comes from CO2. Okay, that's a misconception about plants that a lot of people have. That they get their increase in mass from the soil. No. What are plants? They're a bunch of organic tissue, and a lot of that is cellulose, plant cell walls. And what is cellulose? It's a polymer of glucose. So the mass of all these plants that you see around you comes from atmospheric carbon dioxide. Okay? Now, this particular reaction, we have to reduce CO2 to make sugar, right? We've got to have hydrogens added to it. Where does that come from? The hydrogens come from this reduced NADP. And in the process then, what happens to it? It's reoxidized again. So now this can pick up more hydrogens. Okay? This particular reaction right here is endergonic. So CO2 is down here, glucose is up here. So it's uphill. So we need an input of ATP. Where does that come from? Here. And when ATP is hydrolyzed, we form ADP. And then that goes back and works in the light-dependent reaction again. So those two reactions depend upon one another in order for the whole process of photosynthesis to take place. This is really kind of the guts of photosynthesis. I mean, this is basic information that you should know about the process. And I can pretty much guarantee there will be either an essay question or some short answer questions or some multiple choice questions on the next exam that will ask you which processes happen in the light-dependent reaction, which happen in the light-independent reaction, and then what's the relationship between those two sets of reactions. The rest of it is just digging into a little bit more detail about how all of this happens, and then minor detail into how all of this happens. The Calvin cycle is actually some pretty complicated biochemistry, and we're going to kind of gloss over it so you get the basic idea, but we're not going to dig into all of the gory details of that particular cycle. Okay? All right, so let's take a look in a little bit more detail at the light-dependent reaction of photosynthesis. And here's our chloroplast with the outer membrane. And then the inner membranes consist of these stacks of thylakoid membranes. And I think I pointed this out last time, but it's important to see that these membranes are organized in such a way so that inside each one of these little membrane stacks is a compartment. So we've got a compartment, we call that the thylakoid space. We've got a membrane, and then the second compartment is this stroma. So we've got compartment, membrane, compartment. That's where we're going to set up our hydrogen ion gradient. That's what's going to allow us to make ATP by chemiosmosis. Okay? What's a little confusing is the directionality is opposite the way it is in cellular respiration. So in cellular respiration, we build a high hydrogen ion concentration here in the intermembrane space, and low concentration in the matrix. In photosynthesis, we build up high concentration inside of this thylakoid space, right here, and low concentration in the matrix. So it just kind of is backwards. Okay? So, here is the thylakoid membrane. Okay, this is one of these thylakoid membranes. And embedded into that thylakoid membrane are several different pigment molecules. Most of them are chlorophylls, but we'll see later on that in this cluster of pigments, there are some other what we call accessory pigments as well. Two of the accessory pigments we'll briefly talk about in addition to chlorophyll are the carotenes and the xanthophylls. What color are the chlorophylls? They're green. What color are the carotenes? Orange. They're orange, yeah. Okay, beta carotene is that pigment in carrots that give it that orange color. Xanthophylls are yellow. So these are the pigments that we're seeing in the fall when we have our fall colors. The chlorophyll breaks down first, and these carotenes and xanthophylls persist a little bit longer before they're degraded and the leaf turns all brown. But they're all there, okay? This cluster of pigments that's being shown here, right here, actually consists of 200 to 300 individual pigment molecules. And some of those are chlorophylls, the majority are, but mixed in with this cluster are these accessory pigments as well. These represent kind of a functional unit of photosynthesis. These clusters are called photosystems. And if you look at photosystems in more detail, you'll find out that there are actually two different types of photosystems clustered into these thylakoid membranes. And they are very creatively labeled as photosystem 1 and photosystem 2. So we're going to talk a little bit about the differences between photosystem 1 and photosystem 2. Here is a molecular representation of a chlorophyll molecule. There are actually little variants of chlorophyll called chlorophyll A and B. Don't get hung up on that. The only difference in chlorophyll A and B is a functional group here, an aldehyde versus a methyl group. But otherwise, chlorophyll A and B are almost identical. There's two functional parts to chlorophyll. There's this very complicated ring structure called the porphyrin ring. And right at the center of that is an atom of magnesium. Then attached to that porphyrin ring is a long hydrocarbon tail, if you like. This particular portion right here is very nonpolar. And that's going to be happiest inside of this thylakoid membrane. So this is kind of the anchor, basically, that holds this chlorophyll molecule into this membrane. The porphyrin ring, you'll notice there's a number of places where there are double bonds in these hydrocarbon rings. Double bonds have a tendency to absorb light energy. And remember, only certain wavelengths of light are absorbed by a pigment molecule. Others are reflected. So how many double bonds there are and where they are really determines what wavelengths of light this chlorophyll absorbs and which ones it reflects. And remember, the color you see are the wavelengths that are being reflected. Okay. So if we change this up a little bit, then the molecule could potentially absorb and reflect different colors of light. So chlorophyll A and B will absorb and reflect slightly different colors or wavelengths of light. Okay, little physics now. There is a whole bunch of energy that is constantly bombarding the Earth that is represented by this scale up here. And this scale is called the electromagnetic spectrum. Where is this energy coming from that's bombarding the Earth? The sun. Okay? And what's happening within the sun that is producing this particular energy? Anthony? There's a nuclear reaction that's happening, and it's a special kind of nuclear reaction called fusion. And in fusion, hydrogen is being converted into helium. And when we do that, energy is released. So basically, the sun is a big ball of hydrogen. And under the intense temperature and pressure in the core of the sun, those hydrogen atoms are being forced together to produce a new atom, helium. So is the sun burning itself out? Yeah. We estimate about 4 to 5 billion years before the sun exhausts most of its hydrogen fuel, turns into a big red giant, and then all the planets in the solar system are toast, basically. If all we have to worry about for humanity is 4 to 5 billion years from now, the sun going, red giant, we're in pretty good shape. Okay? There's a lot of other things that are more pressing right now. So this represents the energy that's being produced. This is the electromagnetic spectrum. So gamma rays, X-rays, ultraviolet, visible light, infrared, microwaves, radio waves are all pouring out of the sun as a result of fusion and traveling toward Earth. This spectrum is set up so that these are the shorter wavelengths down here, and these are the longer wavelengths here. And there's an inverse relationship between wavelength and energy. So shorter wavelengths have higher energy, longer wavelengths have lower energy. Which do you think would potentially be more damaging to cells? Shorter wavelength energy or longer wavelength? Shorter. Okay. Higher energy. Some of these forms of energy are so disruptive that they can break up molecules in cells, and they're very toxic. Okay? So gamma rays, X-rays. Ultraviolet rays can still cause problems. They can cause DNA to mutate and cause cancer. Visible light, not so damaging. Less damaging, less damaging, less damaging. And we're constantly being bombarded by these. And at least presently, we don't believe that there is any long-term health effect from being exposed to these longer wavelength forms of radiation. So know where visible light falls in this electromagnetic spectrum. Kind of right in the sweet spot in the middle. Those photons of light have enough energy that they can do some work. But the energy isn't so intense that it damages and kills cells. So it's a very appropriate form of energy to use for photosynthesis. Then if we break visible light down into its color wavelengths, you can see that the shorter wavelength blues down here, and then up into the longer wavelength. I'm not sure why they're showing this gray. These should be the reddish, yellowish colors of light. These are the greenish, yellow colors of light. Okay? So, what happens is, in a chloroplast, visible light energy in the form of photons pass through the atmosphere. And remember that fortunately, our ozone layer filters out and reflects or deflects many of these more intense forms of energy, thus protecting us from them. But the visible light can pass through the ozone layer, no problem. It's absorbed by a leaf. And remember, in the middle of the leaf are these mesophyll cells that are all packed with chloroplasts. And then the light will penetrate into the chloroplast and into the thylakoid membranes. And some of those wavelengths of light will be absorbed by the pigments, by these photosystems, and others will be reflected. It's the absorbed light that does the work. Reflected light cannot do any work in terms of photosynthesis. So this is basically useless wavelengths of light for plants. One of the experiments we used to do in lab, and we've talked about resurrecting this, Dr. Morton and I were just talking about that a while ago, is to take plant tissue and grind it up and extract out the pigments that are in the leaf cells. And then you can take this chlorophyll solution and put it into a spectrophotometer. The spectrophotometer has a light source and then a prism in it, which breaks that light up into different wavelengths. And remember, on the spectrophotometer, you can change the wavelength adjustment. So we use 470 nanometers in our enzyme experiment. But you can set that wavelength dial to basically direct any of these different colors of light through this little slit. And then that shines into the cuvette. And then the cuvette measures the absorbance reading and gives you an output reading. Well, if you do an experiment where you determine the absorbance at different wavelengths of light, you can generate this graph called an absorption spectrum. So what this tells you is the relationship between the color or the wavelength of light and how much light is being absorbed. And remember, it's only absorbed light that works in photosynthesis. We've got three graphs here on this absorption spectrum, and you can see an absorption spectrum for chlorophyll A, for chlorophyll B, and then for the carotenoids or carotenes. And there's some similarities, but there's some slight differences too. You see a big drop-off in absorption down here in these wavelengths. That's because those are the wavelengths of light that are being reflected. The greens, the yellows. Well, chlorophyll's green. The carotenes are kind of a yellowish orange, so they're not going to absorb much. Big absorption spike in the blue wavelengths of light, and then to a lesser extent, another absorption spike in the red wavelengths of light. So plants grab some of the short, high-energy blues and purples and things like that. They reflect the mid-range yellows and greens, and then grab a little bit more of the red wavelength of lights, which are the longer wavelengths. Why the difference between chlorophyll A and chlorophyll B? That one little functional group that affects the absorption spectrum. The carotenes have a very different structure than the chlorophylls, and that's reflected by the fact that they have this different absorption spectrum than the chlorophylls do. So what's the advantage, as far as the plant is concerned, of having both the chlorophylls and these accessory pigments? Why not just have chlorophyll?
Because the other pigments can absorb light from different wavelengths.
Okay, so you can grab other wavelengths of light that chlorophyll can't use. And so you can make more use of the entire visible light spectrum. No, I didn't realize I had that again. Okay. So here's our absorption spectrum. There's another type of spectrum you can do in photosynthetic studies, and that's something called an action spectrum. An action spectrum plots the rate of photosynthesis versus the wavelength of light. Remember, in the absorption spectrum, we're plotting the absorbance of the solution versus wavelength. Now we're plotting the rate of photosynthesis. That word rate again, okay? An amount of something produced per unit time. What's an effective way to measure the rate of photosynthesis? To measure this product, oxygen. If you shine light on plants that are photosynthesizing, particularly if they're in the water, you can actually see the little bubbles of oxygen that are being produced. And you can count how many bubbles are being produced per minute. Well, when you do that, you get a curve that looks something like this. So what does that tell you? At these wavelengths of light, the rate of photosynthesis is high. At these wavelengths of light, the rate of photosynthesis is low. And at these wavelengths, the rate of photosynthesis is high again. Notice how these correlate with one another. The wavelengths of light that are being absorbed by the pigments are stimulating the process of photosynthesis. There was a nifty little experiment that was done quite some time ago. I think this was probably like late 1800s, by a guy named Engelmann. And he did this experiment to generate this action spectrum of photosynthesis using very low technology. And I love some of these historical experiments because the technology that they had to work with was very primitive, but they learned some very interesting things because they were creative in setting up their experiments. So here's what Engelmann did. He took a little filament of algae. This is a photosynthetic organism. And he put it in water, and he mixed in with it some aerobic bacteria. What does aerobic mean? They require oxygen, okay, for their metabolism. And then he took a prism and shined a light through the prism, which broke it up into its different colors, and different colors hit different portions of this filament. And then he looked at the filament under a microscope. And what did he see? He saw clusters of aerobic bacteria on this part of the filament, hardly any here, and more clusters here again. Why? Because these wavelengths of light right here were being absorbed, and photosynthesis was occurring, and oxygen was being produced. These were being reflected, and these were being absorbed. So we got these congregation of bacteria, the aerobic bacteria, where oxygen was being produced. Well, now if we did this experiment, we would have probably a little vernier oxygen sensor, which we could then just use to measure the rate of photosynthesis under different colors of light. But Engelman in the 1800s did not have little vernier sensors. And this was a very creative way to do an action spectrum of photosynthesis. Okay, we've already done that. We did our summary, so we can skip that right now. So, what happens when pigment molecules absorb light? Light has energy. And when a photon of light is absorbed by a chlorophyll molecule, it actually causes some of the electrons in those atoms to become excited, to have their energy elevated. And so when these photons hit a chlorophyll molecule, electrons are temporarily boosted up to a higher energy level. Higher energy, more or less stable? Remember, less stable, okay? So this is the state of instability. So what are those electrons going to have a tendency to do? Drop back down to their most stable state again. That most stable state we call ground state. So what's happening here? Some of the energy of the photons is being converted into electrons being excited. A higher energy level. When they drop back down to ground state again, they release that energy. And they release the energy in two forms. Heat and light. The light that's released is called fluorescence. And in the case of photosynthesis, it produces this eerie reddish color. So if I take chlorophyll in a test tube even and shine light on it, if I could sensitively measure that test tube, it would get a little warmer over time. Some of that energy from the photons would be converted into heat energy. But also when we looked at that test tube, you would see that eerie red glow, which would indicate that fluorescence was occurring. Okay? And start to see what the light's on here. Still can't see it very well, but this has a reddish glow occurring. That is the fluorescence that's occurring. Okay? This is just isolated chlorophyll. Chloroplasts have been broken up, the cells have been broken up, but these pigment molecules can do this all day as long as you shine sunlight energy on them. But has that done anything productive as far as photosynthesis is concerned? No, we're just boosting electrons, falling back down. Boosting electrons, falling back down. Okay, remember that I said that in the thylakoid membranes there are these clusters of pigment molecules called photosystems. In the center, or the heart, if you like, of these clusters, there's a special type of chlorophyll molecule that is called the reactive center. And that's where the really important things happen in photosynthesis, is that this reactive center. So what are all these other chlorophyll molecules doing? And what's their relationship to this reactive center? Well, the other molecules form what is oftentimes referred to as an antenna complex. And they kind of grab the light energy that's hitting the planet. And they funnel that light energy all toward that reactive center. It's kind of analogous to a stereo receiver and an antenna. There are radio signals going all through the air constantly. And if you have an antenna, it's going to grab those signals from everywhere around it and funnel those toward the electronics of your receiver. And if you have the receiver set to the right wavelength, then you can get that particular broadcast source. So this is kind of analogous to the receiver, and this is the antenna. So this molecule absorbs one of the wavelengths of light, and its electrons are elevated. And when they're elevated, they basically collide with another chlorophyll molecule. Their electrons fall back down to ground state, but now they transfer the energy to this one. And that causes those electrons to be elevated. And then as they drop back down to ground state, this one is stimulated. It's kind of analogous to having a series of pool balls on a pool table. And you take that cue ball and you strike it. And when the cue ball hits the next ball, it imparts its momentum to that ball. And it starts moving. And then that hits another ball and it imparts its momentum. And so we get this chain reaction that occurs. So all these things are grabbing photons of light and being stimulated, and ultimately that energy is all directed toward this reactive center. And then an electron is boosted up higher energy level, and something special happens. We'll see what that something special is in just a second. So that's an important concept, is this idea of these kind of antennas out here funneling the light energy toward this reactive center. Okay, here's a little kind of silly mechanical representation of what's happening in photosynthesis. So this would represent one cluster, the reactive center of one cluster or photosystem number 2. And this would represent another cluster of pigment molecules with a reactive center, photosystem 1. And those two photosystems actually are tied to one another. So here's how it works. The worker down here striking his mallet on this little lever that elevates the ball represents the photon of light energy being absorbed by photosystem number 2. So all those pigment molecules, all those antennas are funneling that energy into this reactive center. And what happens? The electron is elevated to higher energy level. But instead of it just dropping back down to ground state and causing fluorescence, there's something there that grabs that electron. It's called the primary electron acceptor. However, if another photon of light is absorbed and another electron is elevated, can this primary electron acceptor grab it? No, it's already reduced. It's already holding its ball. It can't take another one. So if this wants to accept another electron, it's got to donate its electron someplace else. We're calling this the ATP mill. So it throws its electron to this ATP mill. And as this ATP mill is turning, we're making ATP. What is the ATP mill? It's the electron transport chain. Okay? So this electron now falls down to here. Another photon of light, or a different photon of light, is absorbed by this photosystem reactive center. And that elevates this electron up to a higher energy level. And there's something in this photosystem to grab it. This then donates its electron to NADP, and in the process reduces it. So the electrons are constantly going from here to here to here to here. And as a result, we can make ATP, which is needed by the Calvin cycle. We can make reduced NADP, which is also needed by the Calvin cycle. The way this is set up right now, we've got a problem. Because every time a photon of light is absorbed, an electron is boosted and taken away from the special chlorophyll molecule at the reactive center. So after a while, what's going to happen to that special chlorophyll molecule? It's going to lose all of its electrons. It's going to become completely oxidized, right? Remember, lose electrons, oxidized? Now, this particular photosystem could have the same problem, except that the electrons that it loses are being replaced from this photosystem. So what do we need to make this all work? We really need a source of electrons to replenish those that are being lost here. That source of electrons is water. And when water is split up, we produce protons and electrons. Remember, that's a hydrogen. And we produce some oxygen. The oxygen, we know, eventually goes as a waste product. What are the electrons used for? Replenish those electrons that are being lost down here. What are the protons used for? Hydrogen ion gradient. So in photosynthesis, electrons flow from water to photosystem number 2, to the electron transport chain, to photosystem number 1, to NADP. So basically the electrons that are in here came from water molecules. Okay, so here's photosystem 2. This shows you light energy being absorbed by the antenna pigments, one stimulating another, until we happen to stimulate this special reactive center. This has a designation. I don't expect you to memorize that for the exam, but this designation P680 refers to the reactive center chlorophyll molecule in photosystem 2. And when this electron is elevated, it's grabbed by the primary electron acceptor. Water being split regenerates those electrons that are being boosted up here, produces protons for a hydrogen ion gradient, and produces half of an oxygen molecule, basically. But to this point, we haven't made any ATP, and we haven't produced any reduced NADP. So this primary acceptor donates its electrons to an electron transport chain. And we make a hydrogen ion gradient, and now we can make ATP. Remember, by photophosphorylation. But then where do those electrons go? There's no oxygen at the end of that electron transport chain like there is in cellular respiration. But there is another photosystem. Photosystem number 1. Its electrons are also being elevated. So what's replacing them? The ones coming from photosystem number 2. Photons of light being absorbed excite these electrons. Where do they go? They go to NADP, and they reduce NADP. So now we've got our 2 products that we need for the Calvin cycle. And this is our waste product that's being produced as a result of these light-dependent reactions. It's confusing when the first photosystem in the event on the left, because we tend to read left to right, is photosystem 2, and the one on the right is photosystem 1. The association I usually tell students to make is photosystem number 2 is associated with H2O splitting. It's associated with the water. These were really named in terms of order of discovery, what they were discovered. But this always gets a little bit confusing. The first photosystem is 2, and the second photosystem is 1. So this is all of the light-dependent reactions. This is associated with photosystems embedded in the thylakoid membrane. We're reducing NADP. We're using the electrons from water to do that. And as those electrons are being transported through the electron transport chain, we're making ATP by chemiosmosis. And we're dumping off half of an oxygen molecule every time this process occurs. James?
Why do the electrons go to the primary acceptor? Because it looks like the primary acceptor is already getting electrons from the other chlorophyll molecules.
In this one?
Yeah.
It's a one-way flow, and so it's grabbing electrons from that reactive center, and then the water is splitting and redonating the electrons back to the reactive center. So the electrons go here, and then they're boosted here, and then go here, boosted here. So you don't go backwards ever. That answer your question? Ask me after class, okay? If you got a second. We still haven't made any sugar yet. Okay? The Calvin cycle is where the sugar is going to be made. Before we get into the Calvin cycle, and probably it's a good place to stop right now, is we need to dig in just a little bit more into this electron transport chain and see how that hydrogen ion gradient is formed. What contributes to the gradient? Are there different kinds of carriers? Yes, there are. Some are hydrogen and some are electrons. Are those carriers asymmetrically distributed in the thylakoid membrane? Yes, they are. But there's a couple of other events in photosynthesis that contribute to the formation of that hydrogen ion gradient. So next time we'll talk about there's 3 events really that contribute to the development of that hydrogen ion gradient. Then we'll talk about Calvin cycle. Okay?